In this final section of Chapter 17, we're going to take a closer look at how we can see change over time if we really look at the genes of organisms. First thing that we can see is homologous chromosomes exchanging DNA. We've talked about this before. It's called crossing over. Sometimes crossing over isn't equal. So you have an unequal swapping of DNA. One chromosome in that homologous pair gets extra DNA. That extra DNA can carry part of a gene, a full gene, or even a longer length of chromosome. So it's unequal crossing over. Those copies, those extra copies, can undergo mutations that change their function. The original gene is still there, so it's still producing the protein that it needs to. Those new genes, the copies, can evolve without affecting the original gene function or product. So after a gene is duplicated, one of the two resulting genes will undergo mutation. So here's how that would look. We have our original gene here. In an ancestor, there's a copy mechanism that's happened. The mutation in this case has happened to the one on the left here. And then we see a new gene evolving a new function. And like I said on the previous slide, the original gene keeps the original function. So there's still this protein, the original protein is still being produced. This one now is producing an extra protein for a different function. Probably a similar function, but a different protein altogether. Multiple copies of a duplicated gene can turn into a group of related genes called a gene family. And like I just said, the gene family typically produces similar yet slightly different proteins. We're also going to talk about molecular clocks. Scientists use molecular clocks to compare stretches of DNA to mark the passage of evolutionary time. So what we do is we use mutations to mark that time. Neutral mutations tend to accumulate in the DNA of different species about the same rate. We've talked about mutations are random. They just kind of happen. Every so often, an incorrect base pair is put into the DNA strand, into the genome. And in that case, then you see a mutation. So if we know that it's one in every 10 million, one in every 10 million base pairs, then over time, if we see that happening, we can determine if that happens at a certain rate, we can see how long it's been since two species diverged from a common ancestor. So here's an example. You can compare the DNA sequences of two species. Actually, there are 3 species here. And it shows you how many mutations occurred independently in each group. So we have common ancestor. We're supposing a common ancestor. Then we see after 15 million years and after another 15 million years. And here are the 3 different species over here, A, B, and C. So here's the sequence, the original right here. After 15 million years, we see one mutation here and one mutation here. After another 15 million years, now we have two mutations, the original one from here and then another one. Now we have yet another new mutation. So we see a mutation here, a mutation here, and then we see another new mutation here. So because you see that the differences. So we compare these back from these species here. We compare them back and we compare how far removed that original, or these new sequences are from the original sequence. And we use that to determine degree of relatedness. Obviously, we're using neutral mutations because they're not going to change a protein. So this would be, again, sort of in the junk DNA areas of the genomes. So here's the key. The more differences there are between the DNA sequences of the two species, the more time has elapsed since the two species shared a common ancestor. We know because of research that's been done in the past that some genes accumulate mutations faster than others. There are many different molecular clocks that tick at different rates. So we can use those different clocks to time different evolutionary events. As I was saying earlier, we check the accuracy of the molecular clocks by trying to estimate how often mutations occur. They can compare the number of mutations in species whose age has been determined by other methods. So maybe they do some radioactive dating to determine how old a species is or a sample is, and then use its DNA to look at the mutations and calculate how fast that molecular clock is running. Finally, here we're going to talk again about Hox genes. Remember, Hox genes determine body plan, and not only what is developing, but size and shape of what is developing. Changes in Hox gene activity can produce large changes in adult animals. For instance, we have this UBX gene that is present in both fruit flies and brine shrimp. Develops legs. Insects and crustaceans are descended from a common ancestor that shared this gene that had many pairs of legs. But now we know crustaceans still have lots of legs. Insects only have 3 pairs of legs. So in insects, what's happened, a mutation in this UBX gene turns off the growth of some pairs of legs. And this was due to mutations in a single Hox gene millions of years ago. Because of that, modern insects have fewer legs than modern crustaceans. A variant of the same Hox gene, so this would be like the gene family that we're talking about, but instead of in the same organism in 2 different organisms, a variant of the same Hox gene directs the development of the legs of both animals. That's it for Chapter 17.